Yes, thank you for inviting me, and thank you to the previous two presenters.
And if
Fascinating
I can
stuff.
just interrupt, Anna, I would also
Yes,
say, and welcome to Council. For those that sat through the
thank
AGM,
you.
Anna is one of our new ordinary members on Council. So welcome to Council as well, Anna.
Thank you very much. I'm very excited to be joining and getting more involved with spider things. So I've kind of been interested in spiders ever since I've started my uh, education in science. And I was very fortunate to do a master's by research on the Fen Raft spider. So looking at genetics of the Fen Raft spider over time. So that's what I will talk about today. So my first slide, this is a presentation adapted from when I was presenting at Pint of Science, which is kind of like an outreach event for communicating science to non-sciencey people. So this is just adapted from it. So my first slide includes some titles of news articles, quite recent, most of them. Um, so we can see some keywords here, which I'm sure all of you have seen plenty of time. We've got lurks, cannibal, giant, invade, huge sex crazed spiders will invade your house, and how to keep them out. So I think probably many of us why we're interested in spiders is to kind of change people's perspective on that they are not scary, they will not invade your house, they've probably always been there, and that they are worthy of attention and they need more attention and they need more people looking at them and spreading awareness about them. um, it's which brings me to the next slide. This is a very, very simplified food web. So it's all very important, not just the spiders, all the animals are essential for the ecosystems to be functioning properly. And spiders just seem to be the animal that are often unliked and demonized in the media, and people are often really scared of it. So I feel like they do deserve some extra little bit of attention to make sure that they are getting the help that they need. And that is what I did for the Fen Raft spider. Well, just the past year, it was just looking at the genetics. The work for the Fen Raft spider has been going on for a really long time, by probably most of you know, the wonderful Helen Smith, who has been, without her, this project wouldn't have happened. So just a little bit on the Fen Raft spider. It's currently considered vulnerable, and it has quite small fragmented populations, which are seeming to do better now. And it is a semi-aquatic spider, and it's a habitat specialist, so it has quite specific requirements, and it's an important predator in the habitat that it resides in. So it eats a variety of things. It's been recorded eating fish, dragonfly larvae, dragonflies themselves. So they do some important work in the habitats that they do live in. And this is just a summary for the Fen Raft spider from the Spider Recording Scheme website. So we can see at the bottom there in the south, mostly that's their populations. There's I believe 7 of them. So it's very fragmented. So it'll be interesting to find out soon as well, if we can find out why they are so fragmented, because at the moment we are just suspecting that it's because of habitat degradation and habitat loss. But possibly with more genetic information, we might be able to find out why they're so fragmented. So where were we looking? So for my master's, I looked at the population from Redgrave and Loppenfen, which is of the main and best studied populations in the UK, thanks to Helen. And I would like you to notice around, so where you see the dark green border of the trees, that's kind of the area that surrounds Redgrave and Lop and Fen. But if you look out of that area, I would like you to notice how fragmented the habitat is. It's all agricultural fields, so habitat fragmentation is something that these spiders um, are struggling with. So they don't seem to disperse very, very far. And with all of this vast space in between suitable habitat, you can imagine it would be quite difficult for them to move around. Hence, they've been getting so much help with various conservation actions. So this is just a close up. So from far away, if I go back from far away, it can look like there's not much going on, but if you get up close, there is a lot going on in the site. I was very lucky to be able to go out with Helen last year, which was super exciting. We saw plenty of adult spiders hanging around, and it's a beautiful habitat. So if you've never been yet, I would definitely recommend going. There's very well marked trails and viewing platforms where you can see the spider. It's really worth going. It's beautiful there, and it's very rich in invertebrates and in birds and reptiles. So this is just a little bit of a close up of what it looks like. And why was I looking at the Fen Raft spider? So the problem that it's been facing is habitat loss and degradation, like I've mentioned. And then the surveying methods are becoming increasingly more difficult as some of the habitat is not accessible anymore. It used to be, but with vegetation growth, it's really hard to access, and health and safety as well. Some of them are a little bit dangerous to access some of the sites. So using genetic information is another good way to check how the populations are doing, rather than doing visual counts and capturing the spiders. And there's also been a lack of recent genetic work for this species, and just a lack of recognition for spider conservation in general. Um, so a lot of the big fluffy animals get a lot of attention, but as we all know, invertebrates a lot of the time don't get as much attention. So I feel like that's been improving in the recent years with the research that I've been seeing and getting funded, but we still need to push for more to be done. So hopefully that's what I'll be doing. Um, that's what I've done, and that's what I'll keep doing in the future as well. So the hypothesis for the masters was that negative environmental changes in the past have significantly influenced the genetic diversity and structure of the population, and will be reflected by low genetic variation. So how were we able to find this out, whether it's true or not? So first, this is just snippets from when I was doing field work, but the field work has been going on for a very long time. Um, Helen has been collecting the exoskeletons, which I will talk about in a minute. Why the exoskeletons were collected, and this is just yeah, a couple of pictures of the field work, walking around the fens in both Redgrave and Lop and Fen. I also was very lucky to visit the Welsh sites where the spider is present. So yes, very exciting, very rainy, but definitely worth it. And I wanted to show you some of the lab work that I've done in the past year. So these are the exoskeletons, and the reason that we have them and the reason they were collected was to extract DNA from them. So there has been some studies done where DNA has been extracted from exoskeletons of dragonflies, on of cicadas. Not really any that I could find that extracted DNA from spiders. There was a study that used tarantula exoskeletons in Mexico to extract DNA from, but it tends not to be a widely used method because the DNA degrades with time, with the conditions that it's kept in, and it's quite fragmented the older it gets, and because it's just a shed skin, so it's just the exoskeleton of the spider, there isn't really much DNA left there, especially with some of these samples that were 10, 15 years old. It's quite old and degraded, but we still managed to get some DNA out, and we still managed to do some analysis. And yes, which I will talk about soon. I'll show you some of the results. And this is just some of the lab work. I wonder if the videos are going to play. Let's see. Yes, they are playing. Great. So this is, I just wanted to show you a little bit of the lab work that went on. It's just cool to see the stuff that's in the lab. This machine would always scare me every single time. It would scare me so much. It's a homogenizer. So you put in your samples with some steel beads, and they will grind up the sample by shaking it very violently. And in the middle, we have a vortex. So that will just spin around any of the samples if they need incorporating. And then they were meant to stop the video, so I hope it's not too distracting. On the last photo is the PCR machine, which has been around since the late 90s, and it's still going. And that's where my supervisor amplified her samples when she was doing her PhD. And I also used it to amplify my samples. Amplifying means to make lots of copies of the DNA, of the part of the DNA that you want to look at. So that's the machine that was used. It's a PCR machine, so you might have heard the term PCR from around COVID times. So that was used to see if you do have COVID or not. So that would be the same machine, maybe a little bit more modern, that they would be using as well. And let's see, oh, the video's played at the same time. That's fine as well. So this was just me packing up the samples in the lab. So these are the strips that would come out of the PCR machine, and I would get put into tubes and sent off to another lab that would then sequence the samples for me. So on the last photo, you can see the sequences, which was the bane of my life for a few months, looking at those letters, which are the chemical bases of the sequences, which allow us to compare the sequences of all the different individuals. Yes, that they were. It was quite difficult to get my head around it. I've previously not really done any genetic work, any molecular biology, so it was a very interesting experience. But after a few months, I did manage to get some results, which I will share with you now. So we've got two figures here out of maybe 15 that were in my thesis, but I feel like these two figures probably explain what has been happening the best. So what I was looking at is the genetic diversity over time of a mitochondrial gene. So that's the gene that's passed down from the mother, and it always stays the same. And we can see there on the left and the number of sequences against the number of haplotypes. So maybe I should also explain what a haplotype is. So to put it in, to make it a little bit more simple, if my mom passed on her mitochondrial DNA to me, me and my sister would have the same mitochondrial DNA, so we would be the same haplotype. But for example, me and all of the attendees today, we all had different mothers, and most probably those mothers would given us different mitochondrial DNA, so we would all have different haplotypes. So this is just to explain what a haplotype is. So hopefully with the samples that we've gotten of the spiders, the amount of the haplotypes would give us an indication of how much genetic diversity there is. So we do only have half of the information. So hopefully in the future, I'll be looking at both sides of the story. But yes, here on the left, we have the number of sequences against the number of haplotypes. So how many sequences did I have, and how many haplotypes did that give us? And then the years that the samples have been collected over. So we can see that in 1990, the number of haplotypes was quite low, and then it increased. So possibly what happened is that not all haplotypes were collected. So not the samples that were collected didn't reflect the number of haplotypes that were present. And then as we go later down in the timeline, we can see that even though the number of samples was quite high, going up to 15 per year, which still isn't a lot, because there are some challenges extracting DNA from exoskeletons. It doesn't always work. There's only a limited amount of samples that do give us results. So we can see that even though the number of samples was a lot higher, the number of haplotypes still stayed quite low. And then when we get to the end, we can see that there weren't many samples included. So the plan is to include more samples very soon and redo the analysis so we can be sure that we haven't missed any of the haplotypes, and that hopefully the hope is that actually maybe we have missed some of the haplotypes, and we can add that to the results. And on the right, we've got a lovely haplotype network. So it kind of says the same story as the other graph, but it also shows the lines that connect the circles. The colored circles would be the haplotypes, and then the different colors reflect the size and the years of the samples. And then the lines that are connecting the haplotypes and that have lines across them, those represent the amount of mutational steps that would have taken to get from one haplotype to another. And then we have that one little red dot with a white dot in the middle, and that's something that we haven't found. So that represents a theoretical haplotype that should have been there, but we haven't sampled for, that the analysis had brought up. So hopefully, with some more samples added to these samples, we'll be able to get a little bit of a better picture. So the implications of this, these results, is hopefully with more added samples, we can improve the plan for the conservation of the spider. We could possibly think about some new actions to include if it does turn out that the genetic diversity is still quite low. So it gives us a better understanding of the genetic history and genetic diversity of the spider. This framework of looking back in time, although not possible because not always are samples collected so thoroughly throughout the years, is something that could be implemented. So we could be thinking about maybe doing this for other species of spiders or invertebrates that are vulnerable or close to being vulnerable, is starting to collect genetic material over time, over the years, to see how the genetic diversity of these animals is changing. And then lastly is to raise awareness for the importance of invertebrate conservation, which I hope to be doing in the future. So the future involves publishing the work from my Masters by Research, which I've just spoken about today. I have a confirmed PhD scholarship starting in November, which will be looking at population genetics and environmental DNA for the Fen Raft spider, which is very exciting. I'm planning lots more outreach to convince people that spiders are actually very cool. There is another an endangered spider project on the horizon, which I'm sure you'll all hear about soon. And who knows what else the future holds. And now it's time for some self-advertisement. If any of you use social media, if not, it's fine. But I just wanted to also show you what else we do at the University of Nottingham. We have a spider room. It's called the Spider Room. Although most of the things we have are tarantulas, we do have some spiders as well. They're mostly pets for outreach, and we invite people over to come and see our collection of tarantulas and other invertebrates. So if you're ever in Nottingham, there's our email at the bottom. send us an email.
Anna,
And
just one quick question. Your little
yes,
box there is obscuring the email address.
of course. Let's get rid of that.
Perfect.
Right.
Thank you.
So that is the email address. If any of you are ever in Nottingham, you would be more than welcome to come and visit us and to see the lab and to see the spider room. We have plenty of things that can be held, so it's a very nice experience. So we've had some good feedback on it. So hopefully, if you wanted to take a photo of the email or write it down, you have. And this is also, I just wanted to keep this slide in from when I presented at Pint of Science, which I'm sure you all know and you all agree. But I just wanted to keep in the nice photos of the stuff that we have at the spider room and stuff that has been found outside as well. Two of them, a nice little wolf spider and a nice little house spider. So yes, some acknowledgements, mostly to Sarah, my wonderful supervisor, and to Helen, our wonderful collaborator, and everyone else that has been involved with the project. It's been an amazing time, and I'm very excited for the PhD in the future and all the things that we're going to be able to do together. It'll be very exciting. So yes, thank you very much for listening. I also hope I haven't run over too much, and let me know if you have any questions.